Hello, good evening. Welcome to this drop-in class. Just give me a quick wave if you can hear me, or a thumbs up or something, please. Oh, I need to mute this. One second. Okay. Cool. How is everybody this evening? How was the first week of school for anyone in the UK who's gone back this week? Is it nice to be back in the classroom, or are you missing the lines from home education? Alan, you said good for school, or good for, what do you prefer? Cece, you're already sick of it. Yeah, I think people got into quite a nice routine of having a, yeah, you can definitely sleep in a bit later if you only have to be on Zoom by 9 o'clock versus like being in the classroom, which is quite nice. But yeah, you're going to miss your friends, aren't you, if you're not going in. Rita, what exam board is this for? It's for all exam boards. So any of the UK ones, and also if you're doing IB, this is all still relevant as well. Hi, you're new. Oh, nice. I'm sure we've got lots of new people here. Yeah, actually, a quick show of hands. Who's a returning student who's done one of these before? An old hand. Is it the second week back? Maybe. What was last week the first week? I'm not sure. I just do online tutoring, so it's been the same for me. Charlie, Daisy, new, Phoebe, nice. Welcome to the classes. Yeah, so I think I was waiting a couple of minutes to let people give people time to log in, but I think we'll make a start. Oh, Beth, you normally watch the recordings, but you come for the live session this time. That's good to hear. So let me just share my screen with you guys. You should be looking at a PowerPoint now, hopefully. Nice. So, okay, 1 second. The lesson is going to be in an hour today. So it's a 1 hour class, all on mitosis. So yeah, our topic is mitosis. Really important cell division. We need it for, keep meaning to do that update. Yeah, we need it for growth and repair of tissues. So and also asexual reproduction. So one of the core parts of biology is for all specs. It's in there because it's such a fundamental bit. This is AS, but obviously you still need to know it at A2. Yeah. You also have it at GCSE, but you just kind of gloss over it a little bit more. So you go through the basics at GCSE, and then we expand on that in AS. So let's dive in. Oh no, before we do, just a little bit of background information for people whose first time it is. My name is Alex. I'm the head of biology at SnapRevise. I've been tutoring and teaching biology for the last 4 or 5 years. So I used to work in a school in a sixth form college in London, and now doing like online tutoring instead. So a bit about SnapRevise. So we're doing this class today on YouTube to give you sort of a taster of our website. So we've got a subscription package, and I'll talk to you a little bit more about it at the end and sort of give you a demo of the website, but just yeah, some prizes basically that we're giving away. So we're giving away an account today. All you have to do is follow the SnapRevise Instagram, and then you tag your attendance in this class. So just tag SnapRevise with a photo of you and enjoying the class today, and you could be in with a chance of winning it. So we usually get, I think it's around like 100 applications. So you've got like a 1% chance of getting an account, which is pretty good. It's free, so worth doing. We're also giving away a discount code for everybody. So even if you're not Even if you're not a winner, you can still get a discount. So we'll reveal that at the end. So let's dive in. For those of you who've done one of these before, we quite often start with this like learning pyramid, just to sort of revise a few topics that we've done recently. So you'll notice these questions don't necessarily relate to mitosis. It's kind of just some background information that we're keeping fresh in like our students' minds. So we go through the topics, but we keep jumping back because one of the biggest challenges for biology is just learning all the content. It's a massive spec. There's so much to know. So yeah, little and often is the best way for revision. Mitosis, yes. Mitosis happens in your toe. That's a good way to remember the difference between mitosis and meiosis. So anyway, let's jump in. So first question, what is the difference between a scanning electron microscope and a transmission electron microscope? What would you say for that? So it's one of the microscopy is one of the first topics most people do when they do AS. So Rajan is saying 3D, which I like. One uses light. Not quite, Charlie, be careful with that. There's different types of microscopes. So we've got optical or light microscopes, or we've got electron microscopes. They use beams of electrons rather than beams of light. So both of these are electron microscopes, but they're different types. So yeah, for a transmission electron microscope, it looks kind of like the setup. You basically fire electrons through a thin specimen and you get a 2D image. It looks kind of like a light microscope in the principle, but you just get a way, way better resolution because the wavelength of electrons is smaller. They both produce black and white images only, yeah, but the scanning electron microscope is 3D. So 3D image, 2D image. And yeah, the transmission has a higher resolution as well. So the resolving power is greater. And they're still getting better at making these microscopes. So the maximum resolution is always increasing as technology improves. Light microscopes, not really improving that much because we're already at the limit of the wavelength of light. We can't magnify things anymore because literally the wavelength of light is now the limiting factor. So yeah, there's more developments in the electron microscope field. So this is quite a nice one. What's the difference between prokaryotic and eukaryotic DNA? Yeah, nice. Bohemian Corporal, good name. Yeah, so the DNA in prokaryotes is circular, and in eukaryotes, it's linear. What else could we say? Yeah, in a nucleus, good. Don't know why I didn't leave a space there. I've just put in nucleus all as one word, so it looks weird. Whereas, yeah, there's no nucleus in a prokaryote. Very good. Yeah, nice. Introns. So eukaryotic has introns. Prokaryote has no introns. And has anybody, yeah, nice. I was going to ask if anybody's mentioned histones, but yeah, I see a few of you have. So there's no histones in prokaryotic DNA. Whereas eukaryotic DNA has histones. So histones are those little proteins that the DNA wraps around when it coils up. Nice, yeah, and there's also plasmids, yes, has plasmids for prokaryote. Cool. Nice. So next one, lots of good answers there. Next one, what is the internal membrane of a chloroplast called? Could we mention spliceosomes? Yeah, I mean, you're not going to have spliceosomes, or there's no splicing in prokaryotes because they don't need to do splicing because they already they don't have to cut out the introns. Yes, we call it a thylakoid. Very good. Or thylakoid membrane, I guess you could say it was made up of. It's the thylakoid membrane. That's the important one for the light-dependent reaction in photosynthesis. Okay, but that's more of an A2 topic, which some of you guys maybe aren't studying A2 yet, so we won't go into it. Which enzymes are involved with DNA replication? So enzymes, plural. There's 2 enzymes that are responsible for this. Yeah, lots of thylakoids make up a granum, exactly. So we have DNA helicase, or sometimes just called helicase, which breaks the hydrogen bonds between the 2 strands. And we've got DNA polymerase, which catalyzes the formation of the phosphodiester bonds. So makes phospho. Oh, kind of haven't left much space there. Yeah, helicase wants to unzip your genes. That's the way to remember. Cool. What's the enzyme? Bonus question. What's the one that's involved with protein synthesis? So when you're doing transcription, basically making strands of messenger RNA. It's a similar name. Oh, Ramin, this basically ties into your question you've just asked. Yeah. So, yeah, the one in protein synthesis is called RNA polymerase. And actually, there is no helicase for protein synthesis. The RNA polymerase actually does both jobs. It both breaks the hydrogen bonds and catalyzes the phosphodiester bonds. So make sure you're aware of the 2 different polymerases and when they are used. If you're making DNA, it's DNA polymerase. If you're making messenger RNA, it's RNA polymerase. I saw someone mention ligase, or maybe a couple of you. Yeah, ligase is mentioned in some of the specs, but not all of them. I think it's mentioned in OCR, but I don't think it's in AQA, or maybe not Edexcel either. But yeah, that joins the sections of DNA together for the lead and lagging strand. But again, that's not in all specs, and it's not what we're doing today. So I'm kind of just going to gloss over that. So yeah, DNA replication, what direction does it work? Can I explain DNA replication? I mean, we can just do the very brief summary. So the 2 strands of DNA are separated, and then new strands are copied off those 2 strands. So they do it by complementary base pairing with free floating nucleotides, and we call that semi-conservative replication because half of the DNA is the same, is conserved, and half of it is new. Yes, we've had a mixture here. Half the people are voting 3' to 5', and the other half are going 5' to 3'. So yeah, half of you are right, and it is the 5' to 3' camp. So sorry, didn't put a 3 there. So yeah, DNA is replicated in the 5' to 3' direction, and that's because the enzyme DNA polymerase can only bind to the 5' end, so it has to move through that. And yeah, that's why to do the strands, because they run in opposite directions, they're anti-parallel. It's easier to do one strand, the 5' to 3' strand, than the 3' to 5' strand. What does the prime mean? It's to do with the carbon, that the number of the carbon atom that is like facing the end of the molecule. Haven't really got time to go into it now, but it is something you need to know. So that will be worth looking up, Hassan, after the lesson at some point. Yeah, how do you remember the direction? I actually have a trick for this. Just, yeah, one of those things you have to learn. If anybody's got a good way of remembering the direction, then yeah, please feel free to share. I don't know one personally. Okay. Anyway, so I'm gonna just start because we're already like 15 minutes in, so we're just gonna skip over that question. So we're gonna go over the steps of mitosis. We're gonna look at interphase and specifically the gap stages, the G1 and G2. And then I don't think we'll have time to look at binary fission, but if just in case we do, that's the final thing. Crucially, we're not calculating mitotic index today, but that is something you need to be able to do. And it's one of the required practicals for all the A levels. So make sure you're familiar with that. Okay, these are the spec points. Again, I'm just going to skip over them. You guys should all have a copy of your specs anyway. So what are we looking at here? Well, actually, no, it's labeled, so maybe don't worry about the first one. So this is a chromosome. A chromosome is a length of DNA which contains multiple genes. They vary in length. We have 23 pairs in humans. Other organisms have different numbers. So they can have more, they can have less. It's actually, surprisingly, it's not really just to do with complexity, the number of chromosomes. So you might think like a more complex, a bigger, maybe a more intelligent organism would have more chromosomes, but it's not necessarily the case. Some very simple organisms, like a potato, have loads of chromosomes. So a lot of plants have a lot more chromosomes than animals. So yeah, it's not as simple as complexity or anything like that. So what are we looking at here? 1, 2, 3. How would you label these? Evan, some crabs have 300 chromosomes. Really? That's interesting. I didn't realize crabs were very chromosome heavy. Yeah, nice. 1 is a centromere. 2 is, say, sister chromatids. Nice. I've just realized 3 is basically the same thing. It's just saying that they're both identical. Identical DNA. Just so you are aware that they're both the same. So yeah, we quite often see chromosomes as X shapes, but actually chromosomes are just like rod shapes generally when they're by themselves. But when we need to do cell division, so we do mitosis to do cell division, we need to replicate the DNA before we divide, so that when we do the division, both of the new cells end up with 1 full copy of the DNA. So we need to double the DNA, we need to replicate the DNA so that when we divide, we keep the chromosome number the same. It's different to mitosis in that regard. Sorry, different to meiosis. In meiosis, we're creating gametes. So we halve the DNA, we halve the genetic material, plus we reshuffle the alleles a little bit. The aim of mitosis is just to create genetically identical daughter cells. So they have to be exactly the same. So that's why we replicate the DNA. The 2 identical copies of the chromosome are known as sister chromatids. So really important term for this topic, sister chromatids. We use a lot of female and daughter sort of references. So they're called sister chromatids. The cells that you create are called daughter cells from the parent DNA. So noise. Yeah. So chromosomes are supercoiled DNA or condensed DNA, exactly. The difference between a chromosome and a chromatid specifically means one of 2 like identical copies. So this is a chromosome. These are chromatids on the right. So that distinction is important because we need to make sure we're referring to the right thing at the right time. If we're talking about a chromosome, we should say chromosome. If we're talking about a chromatid, we need to be mentioning chromatid. You lose marks if you say the wrong one, the wrong time. So let's go through the stages of mitosis. You guys might know the sort of acronym. So I, interphase, P for prophase, M for metaphase, A for anaphase, and T for telophase. So yeah, just remembering IP mat or what's it called? One of those. What's the name for those rhymes that the first letter is the same? I can't remember what they're called now. But yeah, you can remember IP more after T, which is a nice way to remember the order. Cool. So we're going to go through the stages. We're going to look at interphase a bit later, actually. So we're going to start with prophase. Interphase isn't actually part of mitosis. So only this, the PMAT, is mitosis. It's then followed by cytokinesis. So we could add that on as well. I pray more at the church. Oh, nice. Haven't heard that one. Ariana, the C, yeah, that's the cytokinesis. So you can add that on optionally if you want. Cytokinesis technically isn't part of mitosis, and neither is interphase. I pee myself at Tesco's. Nice. Yeah, I guess there's a limitless number of options. So what happens in prophase with regards to the chromosomes, centrioles, and nuclear envelope? Oh, I party more at the club. You've managed to work in cytokinesis. That's good. Yeah. So DNA condenses. Oh, probably should add DNA. And chromosomes become visible. So when the cells are not undergoing cell division, you can't see chromosomes. They're unraveled. And yeah, they're not visible. They're only visible during cell division. Cool. Yeah, centrioles moved opposite poles. Migrate to opposite poles. So kind of like geography and a globe, we refer to the cell with the same terminology. So either side, these are called the poles, kind of like the North Pole and the South Pole on the planet. And then down the middle, we have the equator. Sometimes this is known as the metaphase plate, but more commonly, we just call it the equator. Put that in brackets. Cool. Nuclear envelope, yeah, breaks down. Exactly. So the nucleus has a double membrane structure around it. We call it a nuclear envelope. That disappears so that the chromosomes can move around. Cool. Can we cover the whole of A-level biology right now? I wish it was that simple, but no. I think arguably biology is the largest spec. I know maybe some history teachers would disagree, but yeah, I think there's a lot to learn. It's hard. It's hard for you guys. I feel for you. So metaphase, what's going on with regards to spindle fibers and the chromosomes? What do I mean by DNA condenses? That basically means that it coils up and becomes visible. Yeah, nice. So spindle fibers are attached to the centromeres. centromeres, and centrioles. So 2 words, don't get them mixed up because they're kind of similar. The centromere is here. The centriole is this one. Very common mistake to get those 2 muddled up because they sound super similar. Happy Mother's Day. I hope everyone has wished their mother a happy Mother's Day, or maybe even got them some flowers or something. Chromosomes are lined up on the equator. On the equator. So they're all lined up, ready to be divided. We've only got 2 chromosomes on our example here, but obviously in a real cell, there'll be a lot more. Cool. Ramin, you're saying biology easier than chemistry. Yeah, chemistry is a hard one as well. I feel like I personally found chemistry easier at school than biology, but I know some people are the opposite and they find biology easier. Oh yeah, what have I written? Linded. Added an extra D in there. Lined up on the equator. Yeah, when I'm talking and writing is surprisingly difficult. Like when you're saying one thing, you end up like writing random letters and words without really meaning to. So nice, that's metaphase. So anaphase, this is probably the most exciting one. Well, not really, it's not that exciting. So in anaphase, there's movement, which is more exciting. So the chromatids are pulled apart. Make sure you're saying chromatids are pulled apart, not chromosomes. The chromosomes get pulled apart during meiosis 1, which we're not going to really go into today. So yeah, spindle fibers contract. You can say contract, you can say shorten, you can say pull, basically whatever you want. It all means the same thing. You can even mention the motor proteins. I think some specs do that. Proteins are involved. I can't remember which one it is. Maybe it's Edexcel. We'll talk about those a bit more. We'll put that in brackets just so yeah, not everyone needs it. Centromeres are pulled. Well, yeah, what were we supposed to be saying here? I guess, yeah, they're separated, pulled apart. Now, this is like characteristic. We have V-shaped chromosomes at this point. So because they've been pulled apart by the centromere, which is like around the middle point of the chromosome, when they get tugged, they form this V shape. So this is a really easy way to identify what stage the cell cycle, what stage of mitosis is going on. If it's V-shaped chromosomes, it's probably in anaphase. Yeah, and they're pulled to opposite poles. Yeah, exactly, Lady Prosecco. Motor proteins pulling on the tubulin. Nice. Cool. Right, final one. So telophase. We've now successfully separated our genetic material. These are going to form the 2 new nuclei. They're genetically identical. They both have 1 full copy of the DNA. So chromatids decondense. You can actually say, it's a bit of a funny one. You can refer to them again as chromosomes now once they're separated. So basically, as soon as they're pulled apart, they're technically no longer chromatids, and you can refer to them as chromosomes again. So when you're saying they're separated, say chromatids are separated and pulled to opposite poles. But after that, you can refer to them as chromosomes again. So I would actually argue that that's the wrong word here. Chromosomes decondense. And yeah, oh no, I think I just looked at my notes. It was saying the chromatids reach the poles. So that's why I was saying chromatids. So then they decondense, so and become no longer visible. Sorry, the order of this writing is now a bit weird. The nuclear envelope breaks down. Nice. And eventually we have cytokinesis. Yeah, decondense is the same as uncoil, exactly. Would you still be penalized? You are running the risk of being penalized for calling them chromatids at this stage. Yeah, I would call them chromosomes. Yeah, nice. So cytokinesis, there's two main ways it can happen. If it's an animal cells don't have cell walls, so they're nice and flexible. They form, basically you get one cell and it's almost like you're putting a belt around a water balloon and tightening it. It gets like thinner and thinner and thinner in the middle, like this. So this would be like stage 1, 2. Finally, we get 2 separate cells. So yeah, it's just like getting thinner and thinner and thinner, and then they basically pinch off and form 2 new cells. Plant cells can't do this because they're not flexible enough because of the cell wall. So what they do is when they've got their 2 new nuclei, they just basically build a new cell wall down the middle between the 2 cells. Now they're separate cells, and both cells can like elongate and grow. So 2 different ways depending on whether they're plant or animals. Yes, the word for that flow is a cleavage furrow. Quite a funny word, this bit. Exactly, nice. The nuclear envelope doesn't break down again, it reforms. Did I write? Oh, yeah, thanks for pointing that out. Why did I write that? Nuclear envelope reforms. And there's now two new nuclear envelopes. Yeah, well spotted, Fredley. Easily done. Cool. So how are you guys feeling with that? Ramin, yeah, you can say chromosome. If one strand is a chromosome, exactly, definitely. Cool. Yeah, cytokinesis does occur in plants. Yeah, that's the one where they form the cell wall down the middle. Nice, mostly 1s and 2s, which is good. Mohammed, Sulat 3, have you just joined, or maybe it's first time through? Nice. So we'll do some questions, and hopefully it'll be a little bit more clear once we've done these. So we've got an image here. We're looking at cells. So this is a practical you guys should all do. You use like, generally it's garlic root cells or onion root cells because they're like rapidly growing. We use the root tips. We squash them and we look at the DNA basically to see what's going on. So you need to be able to identify them. So this one, the student said they are in anaphase, and give 2 reasons to support this answer. So what could you say? Yeah, definitely V-shaped. V-shaped chromosomes is good. and yeah, pulled to opposite poles. Or separated or something like that would be our second one. If you look closely, you can actually see the spindle fibers just about. They're very faint, sort of like all going on here. Sometimes it's more clear than others. There's some really nice like electron microscopy images where you can see it really, really clearly. With a light microscope, this is a light microscope image, it's not quite as clear. The resolution isn't really good enough to see the spindle fibers properly. Yeah, Mohammed, you would be penalized, unfortunately. So yeah, at this stage, once they're separated, refer to them as chromosomes. Yeah, so chromatids are separated. And if you're in the same sentence, you could say they're moved to opposite poles. If you're just referring to them after that, I would say chromosome. Yeah, nice. So let's look at the cell cycle now. So that was one part of the cell cycle. That was mitosis. Actually, wondering why we probably should have done the cell cycle first and then done mitosis. It's a bit of a strange way of doing it, but I guess it doesn't really matter. It's like, yeah, it's supposed to be like a revision sort of class anyway, so you guys should be like familiar with this. They're pulled apart at the centromere. Yeah. They are chromatids when they split. Technically, once they are split apart, they're back to being chromosomes again. So if you're talking about them being split, chromatids, once they're separated, chromosomes again. So yeah, the cell cycle is made up of a few different things. We have interphase and mitosis, and then cytokinesis. So the vast majority of the time, the cell is in interphase. Mitosis is a relatively short part. So why is interphase important? What goes on? Oh, some of you already answered. Nice. Yeah, so growth, DNA, replication, protein synth, all that sort of stuff. So the cell basically needs to do its normal function. So whatever its job is, it does its job while it's in interphase. So people sometimes think it's like a rest state, but it's not resting. The cell is like actively doing stuff. So yeah, we can split interphase up a little bit more detail. So again, this is all interphase. And then cytokinesis would be here. So yeah, mitosis is like the prophase, metaphase, anaphase, and telophase. And interphase is made up of growth or gap 1, basically just called G1, S phase or synthesis, and then G2 phase. So what goes on in each of those things? Cool. Yeah. So cell grows, organelles replicated, protein synth, like basically like normal functions. I saw a couple of you, or at least, yeah, some people are mentioning G0. Yeah, I think it's not in all specs, but we'll just talk about it quickly for those of you who do have to know about it. So essentially, some cells don't actually do like progress They don't go through the full cell cycle. They essentially just stay in the G1 phase forever. So cells that do that aren't actually going to replicate, they just are in G0 phase. G0 is basically like G1, but they're not about to undergo cell division. So they're not like actively replicating organelles, that sort of thing. They're just doing whatever their job is as a cell. Yeah, like a nerve cell, exactly. If you've never heard of G0, don't worry about it. It's not in all the specs. So it was just because some people were mentioning it. If you're looking at that, like, I've never seen this, really, don't worry. I think it might just be OCR that mentioned that. Oh, it does look like go phase, doesn't it? A bit weird. OCR only, I think. How are organelles replicated? Yeah, it depends on the different organelles. Some of them just like split apart and then they can grow bigger. Yeah, some are like built from scratch. It varies. Like the mitochondria, they have their own DNA, so they can replicate. They almost behave like bacteria growing inside the cells. They've got their own thing going on because of the that was their origin. They were endosymbiotic organisms. Cool. So the S phase, this one is DNA replication. So if you're going to do mitosis, you need to replicate your DNA first. So that's very important. And yeah, G2, more growth. We could say DNA is checked. There's a checkpoint here. Checked for errors. This is very important. We don't want to have errors in our DNA. Those are called mutations. And you occasionally get a good mutation. Anyone who's watched X-Men can attest to this. Well, no, obviously not. That's not very realistic. But yeah, occasionally mutations are good, but essentially nearly all the time they're disadvantageous. So we try to avoid mutations coming in. Much more likely to be bad than good, a mutation. Nice. So yeah, checkpoint there. And there's a checkpoint at metaphase just to check that all the chromosomes are also attached. So that's the two that you guys need to know about. Because yeah, you want to make sure your chromosomes are attached correctly so that when they get separated, they both end up with the right number of chromosomes. Deadpool exists. Yeah, he's definitely he's in like, he's part of X-Men, isn't he? He's definitely the best superhero, I think. Or actually, I like, I thought Thor was quite good as well. Hanban G0, basically, it's just, if a cell is not going to be dividing, it doesn't need to progress from G1, essentially. So it's like G0 is like an endless G1 phase. So if it's not actively dividing, doesn't need to do this. So it just stays in like G1, basically. Oh yeah, Wolverine is pretty badass, actually. The first, what was that movie? I think it was just called Wolverine. That was a good one. Yeah, Hugh Jackman. Wasn't he like voted world's sexiest man for like 3 years in a row or something after that film? Cool. Any questions with that? Ready to go through? Just looking at the time. Yeah, we're doing pretty well for time. Let's have a look at this table, because this is a really common way that questions are pitched in exams. So you should be very comfortable with interpreting these diagrams. So they can either have like the mass of the DNA or like amount of like chromosomes or something like that. But so they have this scale on this axis, and then they have time on this axis. And we can use the relative mass of DNA to orientate ourselves with where we are in the cell cycle. So A to B is what? And B to C is what? C to D is what and why? Flo, you don't approve of the table. Guessing you've seen this before then. Doctor Strange. I haven't actually watched that one, but I do like Benedict Cumberbatch, so probably should watch that at some point. Yeah, has anybody got, can anybody work out what's going on in these stages and why? I don't know, I just tried to change these to arrows and they look worse now. Regret that. B to C is interphase. It's a part of interphase, but I've got to be more specific. So yeah, B to C is the S phase of interphase. Of interphase. How do we know it's the S phase? Well, because the mass of DNA is slowly doubling. So we go from 2.5 arbitrary units, doesn't matter what they are. Well, I mean, sorry, they're arbitrary. It just means it's just relative. It doesn't, there's no units. It goes from 2.5 to 5. So from B to C, the relative mass of DNA doubles. So S phase. A to B, therefore, is G1. Nice. And then C to D is both G2 plus mitosis. So we can't tell exactly when G2 ends and mitosis begins, but at some point in this, what's the hours, like between just before hour 8 to just before hour 11, there's both G2 and mitosis occurring. And then at D is cytokinesis. So this is much steeper because it took a little while to replicate the DNA. So we can read off, it takes just under 4 hours for the cell to copy all the DNA in the S phase. But cytokinesis is quick. So yeah, that's a much steeper line. It basically, it splits from having double the amount of DNA to half straight away. G0 it wouldn't look like this. If a cell was in G0, it essentially would like just be a flat line. It's not going to enter the S phase. So it just stays with like a 2.5 arbitrary units of DNA the whole time. Cytokinesis is the separating of the cytoplasms and the two new nuclei that are formed. The word comes from cyto, meaning cytoplasm. Cool. So yeah, on this scale, mass of DNA. So as the DNA mass changes, different, like we can tell what's going on. Yeah, nice. So yeah, hopefully you guys would now all be comfortable seeing those and working out what is going on. So if you want to say like interphase as a whole, actually that's a bit hard to do because it would be like A to somewhere between C and D. So we won't have that. This is basically all we can infer from this diagram. So yeah, and we can also read off like how long is one cell cycle. So this is another way they quite often do ask about these questions. So that they'll show you a graph like this and they'll say, how long is one cell cycle? So to do that, you can just pick any 2 points in the cell cycle, which are the same. So let's go from just before the start of S phase to just before the start of S phase. So we go from 4 to 18. So it's just 18 minus 4 hours equals 14 hours. So one cell cycle One cell cycle takes 14 hours. So every 14 hours, we will double the number of cells if they're growing. Cool. After D is, yeah, no, it's not G0. G0 would just be a straight line. It wouldn't be going up and down because it's not replicating the DNA. So I've never seen a question where they'd like would show on this sort of graph a G01. But I mean, maybe could be a new thing they would do. But yeah, it's not a question that I've seen like that. Okay. Hopefully that's making sense. Just want to do a couple of questions before we finish for today. So let's jump right in. So figure 2 shows the mass of DNA present in the population of healthy cells where mitosis is occurring. So we can see the mass, they're all between 1 and 2 in relative arbitrary units. So explain why some, so let's underline the keyword, why some cells contain a mass of DNA between 1 and 2. What can we say? I mean, 3 because you haven't done this before. Yeah, fair enough. If this is the first time you've seen it, it's going to be quite confusing. Yeah, kind of the pace of these, they are definitely pitched as like a revision pace. So if it's the first time you've seen it, it's going to be a little bit tricky to follow along. Mohammed, what are Golgi vesicles? Very off topic, but they are little balls of membrane that the Golgi produces. Nice. The question is blurry. It's a little bit blurry, but it's not that bad on my screen, so I don't know if it's a connection thing. Yeah. So anything in this one is in G1. In this one is in G2 or mitosis. Anything that's not got in those ones, it must be in S phase. So basically, it's not got just one set of DNA, and it's not got 2 sets of DNA. It's somewhere in between. So all of this is S phase. So for just one mark, they are in the S phase. We can just put that basically. Or DNA replication hasn't finished. Hasn't finished. Okay. Nice. So hopefully that makes sense. Just a one marker, so fairly straightforward. Let's do a maths one now. It's actually quite a straightforward maths one. But let's have a look. So for 2 marks, the time required for a cell to complete the cell cycle was 4 hours and 18 minutes. Calculate the time required in minutes, so important there, not in minutes and hours, for this cell to multiply to produce 8 cells. So we need to work out firstly what 4 hours and 18 minutes is in minutes. Yeah, careful, Samina. You're giving your answer there in hours and they ask for minutes. So it's a sneaky way they have of docking you marks. If you gave the correct answer in hours and minutes, they took a mark off you for not reading the question carefully enough. So yeah, 4 hours, 18 minutes in minutes. So 4 times 60 equals 240 plus 18. So equals 258. So 1 cell cycle is 258 minutes. Now, how many cell cycles we need to go from 1 cell to 8 cells? Well, we can just sketch it out pretty straightforward. 1 cell cycle, we get 2. Oops. 2 cell cycles we get 4. And then our third cell cycle, kind of regretting sketching this out now, actually. 1, 2, 3, 4, 5, 6, 7, 8. We get our 8 cells. So 3 cell cycles. So the calculation is just 3 times 2, 5, 8, which equals getting some very different answers. A lot of people are getting 1032. How, yeah. So the answer is 774. Someone who got 1032 though, I'm intrigued. Talk me through your method. Did you just do like, was that 4 divisions? Yeah. Okay, that makes sense. So yeah, just be careful. Yeah, you just did an extra division. So it's only 3 divisions. Just be careful with that, not 4. Oh, Amy, you just times it by 8. Yeah, no, be careful because it's exponential growth. It doubles each time. So it's not 8 divisions. It's just 3. Cool. Yeah, Kyla, exactly. Kyla, not sure which you go by. That's the calculation you can use. Oh, and Samina's put it as well. Exactly. Nice. Okay, so yeah, that's we're not going to do boundary fission. We haven't got time. So yeah, that was basically our class today. So I hope you found that useful and beneficial. If you enjoyed that, then please do consider subscribing to our website. Just going to quickly like walk you through what it looks like. So going to close this. Discard those. Let's have a look. So this is our website, Snap Providers. We have all the science subjects and maths, and I think we've just launched geography as well. So if you do any of those courses, so let's look at the biology one. Let's look at AQA. So it's a really, really useful package. If you're learning A-level biology, there's really not a better way to learn. So all your topics are filtered here by the different by topic essentially. So you can go through and revise what you want to revise. Let's find mitosis because that's what we're studying today. Oh, we did mitosis and the cell cycle. So let's click on this. So for each topic, what we have is a diagnostic quiz at the beginning. So this is really useful. It tells you how well you know the topic before you set about revising it. So yeah, basically you don't want to be wasting your time revising things that you know well. So the diagnostic quiz will identify what we do or don't know. So I'm just going to click these at random. How many questions do I have to answer? Oh, it was only, wait. I'm not sure if there's only one question or because I got it wrong that can't progress. Anyway, so the bits, so where this is highlighted orange, this orange shows that I don't know this bit because I got that question wrong. So when you get a question wrong, it links to the relevant part of the video with the correct information. So that's like a really useful feature. I think it's unique to Snap Revise. But yeah, super helpful, targets your revision to the bits that you specifically need to know and not the bits that you already know quite well. So you can do this, you do the quiz, watch the video, go through. So we'll just skip to this topic that we didn't know, watch that. And then you can do another quiz at the end to test, or you can do some exam questions and also have a walk through some of the questions with another tutor. Another thing you can do is you can ask questions. So if you're not sure about something you've seen in a video and you've thought about it and it doesn't really make sense, you can post a comment and one of our tutors will answer. So yeah, Millie's asked a question here and Matt has answered her question for her. So really helpful if you're a little bit stuck. We also have these exam packs. So basically like banks of questions for each topic filtered by difficulty. So super useful, again, for when you're revising that topic. Yeah, and with like explanations for each one. So yeah, I think it's a very user-friendly format and really, really helpful. I very much wish I had this when I was doing my revision. We've got some sort of like summary notes, cheat sheets, basically. These are very condensed, all the information you need to know, no waffle. So really useful for just cramming. So I think these are very nice, better than the CGP books, I would argue, with the non-funny jokes, which distract you. The other thing we have is these web classes. So basically, I'm trying to find where they are on the website. I don't usually use the website from a student's point of view, so I'm not particularly good at navigating it. I think it's this way. Yeah, so we have web classes, which basically are like what we're doing tonight, but for a smaller class size. So today there was like over 100 people in the class. On the website, it's like in the region of around 8 to 12 students normally. So you get a lot more. I basically couldn't answer everyone's questions today because there's just too many questions to be physically possible. But in the classes, we do stop and answer all questions. So it's a little bit more interactive. Yes, unfortunately, it is paid. It's a paid service. There's different prices of packages. So depending on what you would like to do. So this is the web classes. So there's one tonight with Tutor, who is running through photosynthesis, so an A2 lesson. And we do 5 a week for each subject. All the old recordings as well are on the website. So if you ever miss a topic, you can just watch it back later. Like here was one I did last week on meiosis. So they're always available whenever you want to do them. Yes, the pricing is, where is the pricing? Packages. I can't find it. There's 3 packages. And is it in here? Yeah, they range from, I think the cheapest one is £10 a month. The most expensive one is £50 a month. You'll be able to find it when you have a look at the website. And it seems a little expensive, maybe £50 a month. It's not free. But for what you're getting, I think it's a bargain. Oh, it's the cheapest one, 20. Okay, sorry. Yeah, you're getting like 5 hours a week of basically like group tutoring. So yeah, it costs around like £50 for 1 hour of private tutoring. So you're getting 20 times more contact hours if you attend all the classes for the same price, and all the other features, the quizzes and everything else. So yeah, £50 a month, but I think it's such a bargain basically for you guys. So yeah, I'll probably I'll stop talking about the website now. Let's go back to the code, because some of you guys are asking about the discount code. So let's put that on. So yeah, remember to set a reminder. Not a very good sentence. Set a reminder for these classes. They run every Sunday. One subject, different subject each week. So yeah, do come along. I'm doing one on photosynthesis in April if you want to join for that. A discount code for £10 off your first month is MIT10. So if you go on the website and you want to sign up, you can use this discount code. There's a free trial for a week. So why not give it a go? If you decide it's not for you, no problem. You can walk away for free. So yeah, and you can do it for £10 cheaper if you do decide to stick with it. And don't forget the competition. One of you can win a free account. And I think that's everything from me today. So thank you for giving up your Sunday evening to learn biology with me and enjoying how dedicated that you guys are. Yeah, I'll stick around for like another minute or so if anyone's got any questions. But yeah, that's the end of the lesson. I hope you learn something and have a good rest of your Sunday evening. Yeah, no worries. You're very welcome. The competition winner will be announced tomorrow. So it's like in the next 24 hours. They'll let you know if you win. Are we considering international boards? Yes, it's on the one of the things that we are working towards. Yeah, I'd also like to see the IB as well at some point, because there's a lot of people doing IB and it would be good to have a thing structured for them. Good, glad to hear it. Yeah, you guys are very welcome. I think we did one on Mitosis actually. Check back through the YouTube recordings. I think we did do a YouTube class on Mitosis. Yeah. Yeah, Mitosis is definitely more confusing than Mitosis. A lot more going on there. How do you sign up for the competition? You need to tag SnapRevise in a post. Full disclosure, I don't use Instagram, so I'm probably saying the wrong terminology, but you have to like tag the SnapRevise page with you attending a web class or something like that. Basically just tag SnapRevise and that's you'll be entered. Nice. So yeah, I think that's probably everything. Yeah, enjoy your evening. Hope to see you guys on the website in the future. If not, hope to see you in one of these YouTube classes in the future. Cool. Bye. Bye.